Hello everyone and welcome at the 21st edition of Science News. Today's story is inspired by an article in Chemical and Engineering News, which in turn reports on a paper in Nature that's concerned with investigating the biosynthetic pathway for strychnine. Strychnine is an indoalkaloid and has been discovered and isolated in 1818 in the seeds of the plant Strychnos nux vomica. It has since garnered a lot of attention from various scientists, especially organic chemists, who have looked at its fairly complex structure, and therefore it became a target for various attempts at its total synthesis, as can be seen on this time scale here. However, it should also be noted that strychnine, or the material from the plant, has been used for a long time before that in Chinese and South Asian medicine. However, and also strychnine has found use in pesticides because it is a powerful neurotoxic, especially in pesticide against small vertebrate animals such as mice and so on. Despite all this, biosynthesis of strychnine has not been properly proved until this point. It has been proposed, but there was no proof of it until this publication. As an indole alkaloid, it was known that the biosynthesis starts from granular pyrophosphate and tryptophan, and essentially up till gessioschizine, compound 1 in the scheme that has been established so far. It has also been known that the violin gumlic aldehyde is involved in the biosynthesis, but the steps from compound 1 to onwards were not properly known. In this study, one of the key ways of elucidating strychnine biosynthesis was the comparison of biosynthetic pathways in the plant that produces strychnine and a related plant, Strychnos sp, which produces a molecule called diaboline. Furthermore, another plant was used, Cataratus roseus, which will provided in its biosynthetic pathways and in its genome further clues in terms of the biosynthesis of strychnine. Furthermore, this work and previous work has established the fact that strychnine and its analogs such as brucine, allostrychnine, and so on are mainly biosynthesized in the roots. This information has been used in this work to focus the work on the roots in terms of looking for the genes of proteins that could be related to strychnine biosynthesis. This has then led to identifying potential genes that could be involved in strychnine biosynthesis in co-expressions analyses of the genes of the pathways that are already known for the initial steps of the biosynthesis that resulted in identification of various genes with potential involvement in this biosynthesis, and again comparison between the strychnine producing plant Strychnos nux vomica and the strychnine non-producing plant Strychnos sp were made as part of this work. The key part of this work then was essentially identifying the potential genes that could be involved in the various steps of biosynthesis of strychnine and their expression in N-beta miana and feeding of these plants containing the co-express genes to see if these plants could be able to produce the various intermediates of strychnine biosynthesis, as is shown here, and the stepwise procedure has yielded insights and identifications of genes and enzymes involved in the various steps of strychnine biosynthesis from compound 1, gasoschizine, to Wieland Gumlech aldehyde, to then onwards to pre-strychnine and on to strychnine. This work has also shown that where the biosynthesis in the strychnine producing plant and the related plant that doesn't produce strychnine diverge is in the synthesis of pre-strychnine, which installs a malonyl group by acetyltransferase, whereas in the plant that doesn't produce strychnine, only an acetyl group is installed to give diaboline. Finally, this work has realized that the final step of the biosynthesis of strychnine involves a spontaneous elimination of carbon dioxide and then the formation of the cyclization. So this is an interesting article utilizing various techniques from genetics that has now given us proper footing in understanding how strychnine is biosynthesized. I thank you for your attention. I hope that you've enjoyed this video, and see you again next time. Take care.